Test, test. I hope everybody can hear us. Welcome to the technical webinar series brought to you by ACEA Biosciences. Today is Thursday for the North American audience and Friday for our APAC region. So good afternoon, good evening, and good morning. My name is Lena Zhao. I'm the Director of Global Support at ACEA Biosciences. I will be the host of today's seminar. It's my great pleasure to introduce the speaker of today's webinar, Nick Mittal. He received his PhD from the MIT in 2010, following postdoctoral training at Mass General Hospital in Boston and the Institution of Bioengineering and Nanotechnology in Singapore. Nick joined ACEA Biosciences as a field application scientist in 2017. In the past 2 years, he has supported and trained numerous customers in the field of Immuno Oncology, who has transitioned from using endpoint assay to label-free real-time xCELLigence potency assay to measure immune cell killing kinetics. The automated xCELLigence potency assay is performed under physiological conditions, thus provide long-term killing kinetic reminiscence of serial killing nature of immune cell in vivo. Add an effector cell to target cell ratio very similar to those observed in vivo. Today, Nick will cover the utility of xCELLigence cell analysis technology using recent examples from the literature, including checkpoint inhibitor, ADCC, cytolytic T cells, including CAR-T, NK cell, etc. The duration of the webinar is 45 minutes. The presentation will take about 30 minutes. Then we'll open the floor to the question, and we'll be providing live Q&A session. Please do submit your question via chat box at any time throughout the webinar. The chat box is located at the upper right of the WebEx screen. We'll try to answer all your questions at the end of the webinar. And also, the webinar recording will be shared with you within the next few days. So, without further ado, I would like to pass the presentation to Nick. Nick, we're ready for you to give the presentation.
Thanks, Dr. Zhao, for the kind introduction. And thank you to all the participants for joining us today for this webinar. So here's the outline for the presentation. I'll begin by briefly describing the science behind our continuous real-time label-free cell analysis technology, namely cellular impedance sensing, and our implementation of it. Next, I'll discuss in greater detail our potency assay for cancer immunotherapy applications. Finally, the rest of the seminar will be run journal club style, where I'll use some examples from the recent literature to illustrate applications of our assay for checkpoint inhibitors, ADCC, and T and NK cell-mediated cytotox. Before we begin, though, just 3 quick slides about our company. We were founded in 2002 and are headquartered in San Diego. In addition to the cellular impedance measurement devices that we'll discuss today, we also make benchtop flow cytometers, including our latest offering, the NovaCyte Quantion, featuring 4 lasers, 25 colors, and 25 independent detectors. Coming back to our xCELLigence line of cellular impedance measurement products, as of this year, we have over 1,500 publications citing our technology. So with that brief introduction to ACEA, let's dive directly into the science. To explain cellular impedance, I have a schematic here showing a single well with 2 electrodes, 1 positive and 1 negative. So in our actual wells, you'll see in a minute, we actually have 30 electrodes per well. So for each measurement, we apply a small voltage of 22 millivolts for only 1 millisecond at a frequency of 10 kilohertz. And multiple studies over the last 10 years have shown that this is perfectly safe for the cells. So in the absence of cells, this voltage drives a small current through the medium on account of its high ionic content. However, in the presence of live cells, this current is reduced because the high lipid content of the plasma membrane and the fact that lipids are poor conductors. So you may recall from your high school physics that impedance is defined as the voltage divided by the current. So as that current decreases, the impedance increases. So in our actual system, we displayed the background subtracted impedance value, which we call the cell index. So for example, here's a typical cell index plot for HeLa cells plated in one of our wells. And you can see that as the cells divide and cover more and more of the electrodes, you see that exponentially increasing the cell index, as one would have expected. In terms of our implementation of this principle, the heart of the technology is what we refer to as our E-plates. As shown here, the bottom of each well contains 30 interdigitated gold microelectrodes, which have our patented string of pearls design. The key feature of this design, as you can see in the bottom image, is that the electrodes cover 80% of the bottom area, providing the maximum possible sensitivity. In terms of the plate readers themselves, we refer to them as stations. So all our xCELLigence stations are designed to be placed in regular tissue culture incubators. Our three models shown here are the SP on the left, which runs a single 96-well plate. The MP system, multiple plate system, can read up to 6 96-well plates, and our DP system, which can independently run 3 16-well plates. Finally, as shown in this lower image, all the systems connect via a flat ribbon cable to the analyzer and control unit that sit outside the incubator. Briefly, we also offer an HT system for working with 384-well formats, as well as a specialized instrument for working with cardiac myocytes. In terms of applications, xCELLigence technology can be used to study a wide variety of cellular processes. Of course, today we'll focus on cytotox, but one way of thinking about it is that just as a microscope interrogates your cells with light, our system interrogates the cells with a small electric current. So just as a microscope can be used to investigate multiple aspects of cellular physiology, impedance can also be used to investigate multiple aspects of cellular physiology, with the added advantages of being completely label-free and greatly simplifying the data analysis. The key advantages of impedance measurements relative to other types of readouts are as follows. Number one, it's a non-destructive measurement. So this means that one can get repeat measurements or complete kinetics from just a single sample. And additionally, one maintains compatibility with orthogonal assays. Number two, it's completely label-free and therefore more physiological relative to having to add dyes or labels to one's cells. The temporal resolution is very high. It can read as fast as 15 seconds for a 96-well. And finally, it's really easy to use both in terms of operating it as well as the data analysis. So with that introduction to impedance sensing, let us focus now on our potency assay. The central challenge for any potency assay for Immuno Oncology is that we have a mixture of cells, and we selectively need to isolate the survival of the tumor cells while excluding from the immune cells. Our assay is differential adhesion to immune cells. First, let's look at the example. Cells attach easily to our gold biosensor than index much greater than 1. If you look at most of these T cells and NK cells, these are non-adherent, and so they typically produce a low cell index, typically less than 0.1. So there's usually an approximately 10-fold or greater difference between the cell index of the two cell types. And so when we mix the two cell types, and then we observe the decrease in signal corresponding to cytolysis, then we can be confident that the majority of that decrease is really coming from the tumor cells. Additionally, for non-adherent tumor targets, we've developed several tethering reagents that can selectively bind these to our gold biosensors, and we'll describe these in greater detail shortly. Perhaps it's useful at this stage to briefly compare our assay to other commonly used potency assays. One commonly used class of assays is to selectively pre-label the tumor cells. So this can be done with a passive label or dye such as chromium-51, or alternatively an active enzyme such as luciferase. Relative to both these types of assays, our assay has the advantage of being completely label-free, of course, and additionally providing continuous monitoring because of the non-destructive nature. So this enables the capture of the non-linear nature of cytolysis, which is a topic we'll return to shortly. Further, with regard to chromium-51 in particular, our assay offers the advantage of not needing to work with radioactive materials, and it also enables longer assays, which in turn enables the use of lower and more physiological E/T ratios. With regard to assays such as luciferase, additionally, while in principle luminescence can continuously be monitored within plate readers, typically lab plate readers may not provide full physiological conditions, that is, the temperature, CO2, and humidity. So on the other hand, since our stations are housed within your tissue culture incubator, the measurements are obviously made under full physiological conditions. Another commonly used assay is to trypsinase stain and FACS cells. Relative to this assay, Impedance provides the advantage of reduced handling, and in addition to just saving you time, this also reduces the variability associated with such measurements. Last but not least, imaging is also commonly used to monitor potency. And here again, our assay offers the advantage of being label-free, and additionally, the data analysis you'll see is greatly simplified since there's no training of the software, etc., required for our analysis. So the remainder of the seminar I'll run in a journal club style, so I'll give some examples from the recent literature where our assay was used to assess potency. And I wanted to mention that while I'll focus on immune regulation, ADCC, and T and NK cell cytotox, our system can certainly be used to assess the potency of any other type of cytolytic agent as well. The first paper I'd like to present is a study that was published late last year in Cell Reports, and this paper investigated a novel role for PD-L1 in cancer. So the role of PD-L1 in diminishing T-cell cytotox is well established. But here they focus on another role of PD-L1, which is in counteracting interferon-mediated cytotox. So let's begin with this figure where they show the effects of antibody-mediated PD-L1 blockade. Here they're using the B16 mouse melanoma cell line that expresses PD-L1. So shown here on the left is the impedance data generated with one of our instruments. The red trace is showing the isotope antibody control. So even in the presence of IFN beta, due to the endogenous expression of PD-L1, these cells indeed can continue to grow. However, as shown by the other traces, in the presence of increasing concentrations of a previously validated function-blocking PD-L1 antibody, one can clearly see the dose-dependent increase in the cytolysis of these cells, and then the endpoint data is captured in the bar graph to the right. Further, they expand their repertoire of target lines and show similar effects in other murine and a human cancer cell line as well. They complement their antibody approach with genetic experiments, as shown in this figure. Here, let's focus on these two traces. So the red trace corresponds to a PDL overexpression cell line, whereas the blue trace corresponds to a B16 PDL1 knockout line. So here again, the xCELLigence data clearly demonstrates that PDL1 offers protection from the cytotoxic effect of IFN beta. And additionally, they dissected the roles of various intracellular domains of PDL1, though we won't go into those details. So let's now move to their in vivo data shown in the second half of the study. Briefly, here's their model. They inject B16 cells subcutaneously. Then after 7 days, they perform an intratumoral injection of a lentivirus that drives IFN beta expression or a control that drives GFP expression. and then they followed this by another injection after another week. So here's their data. The left panel shows that relative to the lentivirus that drives GSP expression, the lentivirus driving IFN beta production indeed leads to a decreased tumor size, as expected. Now in the middle panel, they're showing the results for the B16 line that stably overexpresses PD-L1. And so what we see is that this line is no longer sensitive to IFN beta. And finally, the right panel shows data for a stable modified B16 line where now PD-L1 has been knocked down using short hairpins. Importantly, what we can see is this line was far, far more sensitive to IFN beta. And in fact, in one tumor, you can see here that the tumor was almost completely lost by day 32, though it eventually grows back. Similarly, with PD-L1 knockout cells, the tumor was reduced, especially in the presence of IFN beta, and one can easily see that those mice lived the longest. So indeed, the in vivo data correlates very well with the in vitro xCELLigence data. I also briefly wanted to share this data from Judy Young at Genentech, which illustrates that our platform can be used as screening for potency, but also for specificity. So here, as you can see in the top panel, cells expressing the target antigen display clear dose-dependent decrease in cell index, whereas cells in the bottom panel that do not express the target antigen clearly do not show this type of response. Further, multiple clones of the antibody could rapidly and efficiently be screened. And last but not least, I wanted to again point out that the traces here clearly demonstrate the nonlinear nature of cytolysis, and we'll come back again with an even better example in a couple of minutes. Next, let's move on to T-cell mediated cytotox. The first study we picked here is a recent study from the Clinical Center at the NIH. This study focused on the manufacture and QC of clinical-grade GCR transfused T cells. In the introduction, one of the interesting things they point out is that while typical CAR T cell protocols require 1 to 5 billion cells, GCR transfused T cell protocols have typically required much higher numbers of cells of the order of 100 billion cells to obtain clinical responses. We won't focus here on the details of the manufacturing process, which are of course provided in the manuscript, but we should just note that the T cells here were transfused with TCRs for either the E6 or E7 HPV oncoproteins, and also that the process maintained high viability in both these cell types. So the data shown here right now is for the E6 TCR, but similar results were obtained for the E7 as well. Let's focus now on their potency QC data, which were performed using an xCELLigence MP system. The unity used here was 1 to 1. So here, as shown in the first bar, for the Cas9 line, which is known to express HPV viral sequences, and to which the donor T cells were HLA matched, they observed cytolysis. On the other hand, for the CHA line, which is HLA mismatched now, cytolysis was not observed. Similarly, for the 293 T cells, transfused with HLA-A2 and E6, they undergo cytolysis, while those that had a mismatched antigen, so namely they were transfused with E7, cytolysis was not observed. So this demonstrates both the potency as well as the specificity of these cells. And similar results were obtained for the E7 TCR transfused cells as well. This next study I'll touch on just briefly. This one is from the bioelement lab at the Baylor College of Medicine, where they've developed novel CAR constructs with multiple domains within a single receptor. So these can then target multiple antigens. Another point that's worth noting is that they use a very low concentration of effectors in their assays. So the effectors are only 5% of the total cell number, which is similar to what is observed in vivo. In this study as well, they subsequently observed excellent agreement between the xCELLigence potency assay results and the results from their mouse model of TBM. This data also raises another very important point. So look at the in vivo cytolysis curves here on the right side, and you can easily see that these are highly nonlinear, and the mechanism here is thought to be effector exhaustion. But as we saw earlier with the data from Genentech, and here's some more similar data from Nabil Ahmed's lab again. Here, this shows various CAR-T lines monitored in vitro on an xCELLigence SP system. So what you see here is that by running these assays for these longer times, it's 1 week for this particular data, and with these low physiological EDTs, it appears that it's possible to recreate this phenomenon of effector exhaustion in vitro as well, and especially for the traces highlighted here with the black arrows. One final paper I'd like to briefly discuss is a recent publication from Carl June's lab. Here they evaluated a combination therapy with an oncolytic virus that also produces TNF-alpha, and they're doing this in combination with CAR-T cells targeting mesothelin. So as shown here, the CAR-T cells alone, as well as the virus alone, certainly cause cytolysis. But certainly in the presence of both, there's a strong synergistic effect as shown by the red trace. We'll conclude with a couple of examples of NK cell-mediated cytotox. Here I'll highlight data from a paper that we recently published this year in PLOS One. In these experiments shown here, the NK92 cell line was used as effector, and effective cytolysis of both the PC3 prostate cancer line as well as NCS7 breast cancer cells can easily be observed here. Additionally, for non-adherent cells such as B-cell lymphomas, we have developed targeted tethering reagents that selectively bind the target to the center while excluding binding of typical effector cell types used in such assays. Dose-dependent cytolysis is easily observed for these targets as well. And further, with the aid of simple formula, the normalized cell index can be converted to percent cytolysis, and these values can be compared to those obtained, for example, by FACS. So here we show xCELLigence data for multiple EDTs, shown in blue, agree very well with the FACS cytolysis data shown in red, at both 4 hours, which is shown on the left, as well as 24 hours shown on the right. So this table shows some of the other cell lines that we currently have tethering reagents available for, and is available upon request. Finally, one can certainly also use primary NK cells as effectors, as was demonstrated in the study from ISRM. A final thing I'd like to discuss, and this spans across all classes of therapies, is the option to run assays with patient samples in our system, which enables personalized medicine. We have our own ongoing project in this space in collaboration with MSKCC, about which you can read more on our website. However, as this is a relatively recent and ongoing project, we're unable to share any data from this particular project at the moment. However, to illustrate the concept, I'll use the study published last year in BLC Cancer. Here they start by observing that previous clinical studies have suggested that a subset of HER2-negative patients still respond to anti-HER2 therapies. So their motivation was to see if they could develop an assay for defining the subset prospectively. So currently scoring of HER2 positivity is done via IHC and FISH, which look at expression, so they can't help with this problem. So they wonder here if they could instead use a functional assay. To understand their assay, let's briefly look at the biochemistry of HER2 first. So HER2 is the second member of the HER family of receptors, also known as ERBD, and currently there's no known ligand for HER2, but it is known to dimerize with the other receptors in the family, and some of the ligands for the other family members are indeed known, for example, EGF and neuregulin-1. So to test the response to these ligands, they used a commonly used HER2 positive line, SKBR3, in our system, and observed that upon addition of either EGF or NRG1, there was a strong increase in the cell index over the course of approximately 30 minutes, likely due to increased adhesion of the cells to the plate. Further, they tested the effects of a few inhibitors. The main one they used is pertuzumab, so let's focus on that one. So pertuzumab is an FDA-approved drug that blocks the dimerization of HER2 with other HER receptors. So the next set of experiments, they pre-treat with the drug. So you see that it has no effect in the absence of the ligand. Now in the trace shown here, after 18 hours, they added NRG1. So here shown in the pink trace is the activation observed with NRG1 alone. However, with the pertuzumab pre-treatment, as shown in the red trace, this activation is greatly decreased. Additionally, for example, they can easily develop IC50 curves for both the neuregulin as well as the ETF-driven signals. Based on these observations, they then developed a metric of functional HER2 activity that they call the total HER2 signal. So while the details of the metric are proprietary, what they do say is that it is based on the reductions in the NRG1 as well as the ETF signal produced by pertuzumab, as we saw in that previous graph. So they then went on to test a panel of 19 breast cancer lines. 10 of these were HER2 negative and 9 were HER2 positive based on expression. And they did say that the gene as well as the protein expression data were consistent for this set of cell lines. So they then measured the functional HER2 signal for these 19 lines. And these two observations seem the most interesting. So first off, there's one line that is HER2 negative by expression, this BT483 line here. But interestingly, it actually showed the highest HER2 activity of all the lines in their assay. Secondly, there were 4 lines that were classified as HER2 positive based on expression, but they turned out to have activity in their assay that is similar to several of the HER2 negative expression lines. So additionally, they ran a correlation analysis, and indeed they find a very poor correlation with R-squared less than 0.1 between their HER2 readout and HER2 expression levels measured by FACS. Here's the last figure now. So here they show data for actual patient samples and controls. So here's the SKBR3 positive control. The first black bar is the signal with neuregulin alone, and the second shows the decrease in that signal with pertuzumab. Correspondingly, the last data here is for the negative control cell line MB231. And then here's the other negative control. Sorry. So here's the other negative control, which is the healthy patient. But what's interesting is that they then have 2 patients here that are HER2 negative, and you can see that both, that for this R39 patient, indeed there's a high HER2 signal, but for this other patient, R49, who's also HER2 negative by expression, there's a very low HER2 signal in their functional assay. So this is something that they're now going to probe in a clinical trial. So in conclusion, the key advantages of impedance measurements are as follows. Number 1, it's non-destructive. Secondly, it's completely label-free, which in turn enables longer assays at physiological unities, the high temporal resolution, and last but not least, the ease of use, both in terms of operation as well as data analysis. One final item, we'll soon be launching our RTCA Pro software. This will have a number of features for immunotherapy applications, for example, automatic conversion of cell index data to percent cytolysis and other such features. So if you'd like more information about this closer to release, please leave us a note or send us an email, and we can add you to our mailing list for this. with that, I'd be happy to take any questions. Thank you.
Great. Thanks very much, Nick.
Thank you.
Very good presentation. So now I'm going to compile the question that has already been submitted during the seminar. The first question, I guess I can answer that. Will the presentation be available? Yes, the presentation slide deck and webinar recording will be shared with the participant in the next couple of days. The next question for Nick, is it possible to isolate and probe against the effector cell after the experiment to monitor serial killing?
Sure. Yes, that's something we've tried and something that can definitely be done. So because our assay is completely label-free, you can certainly go ahead and pull out the effector cells and reprobe them. And in fact, you can also, reprobe or do other types of assays, as we mentioned, with the tumor cells themselves. In fact, here's a slide. Let me just pull it up. Just a second. Sorry. So as we showed in our paper, for example...
Nick, can you share your screen one more time?
Yeah. Just one second, sorry.
Great.
So as we showed, for example, in our data from our paper, where we compared RTCA with flow cytometry. So that's just a quick example where we're able to pull out those cells and then analyze and fax them after staining. So for example, that's one type of thing you can do. Additionally, if you wanted to image the plate, we also offer these view E-plates, as we call them. So in these plates region, 4 electrodes have been taken out, which really does not affect the impedance performance, but it does enable enhanced imaging. And you can, of course, also stain the cells, so crystal violet staining. So you can also do imaging type assays. And then last but not least, you could also certainly do biochemical assays if you run MTT, flow, and so on. Certainly possible, or lysosomal RNA, and so. And finally, what we'll like to do is kind of cytokine analysis. And certainly, so you can always pause the experiment, even though, say you have the data every 15 minutes, collect some of the supernatant cytokines, and then continue collecting the impedance data.
Thanks. This is great. It already answered quite a few questions that has just been submitted asking whether or not you can sample the cytokine secretion and then how does our data compare to conventional assay like imaging analysis. Thank you very much for the very complete. The next question is, it reads, is the technology in a mixed target population? I guess they want to see how low the effector cell to target cell to be able to observe any cytolysis effect in our.
Sure. So in terms of the E to T, if you look at that study, for example, from Baylor, they're using an E to T of between 1 to 10, or what, by that I just want to clarify that means out of the total mixed population, the effectors are only about 5 to 10% of that total population you mentioned earlier as well. The other way activity potentially is in terms of the amount of killing that can be captured. Our wells, typically the sensitivity, of course, it's a little bit cell type dependent, typically right around 100 cells or so. You can imagine if you have a typical target population of about 5,000 to 10,000 cells and of about 100 cells. So that comes down to about a 1 to 2% change in cytolysis can easily be detected.
The next question is, what is the longest time you can run the assay for?
Sure. So the length, for example, the data from Ahmed, that was for about a week. That can be done, and really the only limiting factor is how often the cells and so on. So for starting to run out of nutrients, one can sort of do medium exchange, and that way you can in a cardiac system run assays even for 2 or 3 months.
Great.
And the cells seem to do just fine.
There is one question. Can this assay be used to probe macrophages?
Yes, absolutely. So yeah, this tends to be an application, which is why we didn't studies as such, but certainly that we can send to you comes to mind is from and David Pantoja's lab at Wake Forest.
Thanks.
So we'd be happy to send that across.
I have 2 more questions. Probe immuno, and I think how you have shown the T cells study, and also another study that you've shown as example.
Right. So actually, we presented toxicology for that poster along. Focused on immune toxicity. The way we approached that was we had a specific cytolysis to see, for example, the cross-reactivity to have immunological effect.
The data using your RTC.
Really, Excel figures, of course, if you just want a quick data, but in terms of the actual data, we export Excel, and that's the data as well, any data analysis, all this, just a couple.
So, anybody will, I believe you, is this a B-cell killing assay?
Right, this is, so flow cells in our study, the publicly available. But basically, we find that the stage of cytolysis combination, for example, a combination of the to get really a match between.
So the B-cell killing, either cells with cell for RTSM, the result of shown here. Yeah. Okay, so I think all info at our tech. One topic that you would like to hear from us, get your input and in the webinar. Also our audience of your busy schedule minutes, and have a good day for colleagues. All right, see you next time. Bye now.